All right, so last time we looked at actin, we looked at myosin. Myosin is the bigger word, it's the thick filament. Again, its shape, one myosin protein, it has two tails that are squiggled around each other. Don't care about the tails. I care about the heads. Again, it's the business end of the myosin. It's where things happen. That's why we call it even called, there's active sites. It's where things are happening. The main things that happens at the head of the myosin, ATP will bind there, and it will bind to actin, that thin filament. And so we care about what's happening at the head region of the myosin. The thin filament, the actin, also has two twisted proteins to it. Now again, they look like little blueberries that are all twisted together, and I can never draw this very well. Tropomyosin, and I'm like, covers all those little dark circles on there. Those dark circles are active sites. As I said before with myosin, myosin will bind to actin, and it will do that at those dark circles, the active sites. So a myosin head wants to bind to that dark circle right there. However, is the dark circle available to be bound to right now? No, because it's that tropomyosin, that orange protein that covers it. And I'm like, so we can't always have muscles contracting, otherwise they're always just contracting out of control. And so we cover all of those active sites on the actin with that tropomyosin. Now, troponin's job, the yellow structure, is just to anchor that tropomyosin in place. Like, hey, your job is to cover all those active sites until we move. And so nothing's going to happen. No muscles will contract. Nothing will happen if those active sites are covered. Now, my goal today is to hopefully get through far enough where we will go through how does a muscle actually contract. And yes, it will end with the myosin heads with their actin binding sites are going to bind onto those dark circles, those active sites, and they will grab on, and then they'll try to shorten the muscle. Now, looking at a bigger picture of it, if we've got that myosin, the thick filament, you can see all the little heads sticking out. Again, it's all the little heads. We don't care less about the tails. We care about the heads. Those heads, all of those heads on the thick filament are going to grab onto the thin filament. I'm like, if those active sites are open and available, all the heads are going to grab on to those active sites, and they're going to shorten the muscle. Now, some different structures that we find on our myofibrils, and I'm like, these are things inside of our muscle cells. One, we have elastic filaments. They hold the thick filaments. They hold the myosin in place. Also, they help with recoil and stretch. And I'll show a picture of it. They actually look like a little coil. So when they stretch out, when you think of a little something coiled, it likes to pull back. Like one of my pictures. Here we go. And I'm like, here are those elastic filaments attached to the myosin. And so when the muscles stretch, because that little coil, it pulls it back. There's also dystrophin, is a protein that links the thin filaments, actin, to proteins found on the sarcolemma. What's the sarcolemma? What's sarco mean? Or maybe someone said it. Muscle? Lemma? Membrane. It's the phospholipid cell membrane, but on a muscle cell. And so we want to link those actin filaments to the actual membrane of the muscle. I don't know if I had that picture on here or not. Nope, it doesn't have the dystrophin on there. Now, different structure, a unique structure that we find in the muscles. It's called the sarcoplasmic reticulum. Again, sarco means muscle. It's on a lot of these words. It is a network of smooth endoplasmic reticulum. So it's like smooth endoplasmic reticulum, but it's found in muscles and it's got a special job. So it's kind of like an extension. This is all found in cells. Now your sarcoplasmic reticulum is all of this blue. It's all of this blue structure that's on here. That's your sarcoplasmic reticulum. Most of it runs lengthwise with the muscle cell. However, at the ends, and I'm like, where it comes across this little white structure called a tubule, a T-tubule, and I'm like, it now runs top to bottom. It has these little perpendicular ends. Now, the ends, we call them terminal cisterns or terminal cisternae for plural. It just means you have an end here and an end here. There's an end here, runs up and down, and an end here, up and down. They're the terminal cisterns. They just form perpendicular, up, down, versus running lengthwise. Now, the function. Know the function, highlight the function. The function of sarcoplasmic reticulum is to store and release calcium. What else in the body stores and releases calcium? We had a whole unit on it. Bones do. And I'm like, sarcoplasmic reticulum also stores and releases calcium when needed. Because yes, you know, if it needs more calcium, it will get it from the bone. Now, we also have T-tubules. And I'm like, so they are tubes. They're the white structures on here. They are continuous with the sarcolemma, with the muscle membrane. Like if I had a big old muscle, long muscle cell right here, and it's all covered in the sarcolemma, the muscle membrane, that phospholipid bilayer. And I'm like, I could take it and dip it in a little bit. And I'm like, and I form these little tubes. It's almost just like I sat there and I'm like, this was attached to the outside of the muscle, and I just sat there and, you know, divvied it in a little bit. One, it gives lots of surface area, but now things that were on the outside of the muscle are now inside the muscle. Again, because we're going to have to send information inside. So we have lots of surface area now, and we find the tubules on either side of those terminal cisterns, the ends of our sarcoplasmic reticulum. So you always have a sarcoplasmic reticulum, T-tubule, sarcoplasmic reticulum. Three things. So we call it a triad. So two terminal cisterns, the ends of the sarcoplasmic reticulum, and a T-tubule in the middle. There's three things that always come together, which is good. And I'm like, because you're going to see this again. We have information on the outside of our cell. It will come inside the cell through T-tubules, because it's all continuous. Comes inside the cell into a T-tubule, and it will tell these sarcoplasmic reticulums to release calcium. So I'm like, it works out well that T-tubule is right in the middle. There's a bigger picture. Yeah. I was going to say, oh, I like this one. Because there's a sarcolemma, the muscle membrane, and it just dips right in and now becomes part of the T-tubule. Now, the sliding filament model of contraction. This is how our muscles contract. Those filaments, your actin, your myosin, can slide past each other. The heads will grab and they move the filaments so that they shorten. Now, it doesn't mean they have to shorten to generate force. I'm like, if I sat there and I'm like, here, we'll just use my wall over here. I'm pushing on the wall, kind of hard. And I'm like, the muscles that are pushing right now, are they moving? Nope. However, am I generating force against this wall? Yes. And I'm like, they don't have to shorten. You know, I can generate force against something. The muscles don't have to shorten. It just means the myosin head attached to actin, and it's got lots of force holding on to it. You know, it's only when we shorten it, you know, I can sit there and I can move stuff. I can't move this wall. It'd be cool, but I can't. And I'm like, but that doesn't mean I can't generate force against it. The shortening, when the muscles actually shorten and you can see movement happening, only happens when, yes, there is a tension or force, and I'm like, greater than opposes the shortening. Fancy way of saying, I'm stronger than the wall is. And I'm like, my force is greater than the tension that comes back from the wall, and I could push the wall. I can't. And I'm like, but, you know, I could have. It was strong enough, or I tried to move something smaller or shorter, or didn't weigh as much. Now, what we're talking about sliding filaments. Again, we have thin filaments, my little blue lines, thick filaments, my myosin. This is a fully relaxed sarcomere. Means these are those repeating units. Here's one sarcomere, one would be over here, one would be over here. And I'm like, this is relaxed. My thin filaments are pretty far away from each other, and the myosin's right in the middle. However, if I contract and those myosin heads grab onto them, they're going to sit there and they're going to do this, and the actin as they do this is going to get closer and closer and closer. And so those blue lines, which were really far away from each other, are now almost touching each other. And so this sarcomere has now shortened. That means like really long, shortened. You're not talking this muscle. Nice and long, shortened. And I'm like, when that happens, you just have these little myosin heads that just grabbed onto the actin and moved it closer and closer and closer together. And this this little sarcomere shortened, the next one shortened, the next one shortened. They're all shortening at the same time, and now your muscle is shorter, and you see movement happening. I was going to say, I'm like, oh my God, yes, actin and myosin, they overlap. And I'm like, anytime a myosin attaches to the actin, we call that a cross bridge. It's just the attachment site. So I'm talking about cross bridges. I just means the myosin head actually attached to the actin. The active site was open, it attached. It formed a cross bridge. It formed an attachment there. This I'm like, I don't think I have audio, which is fine, because you can still just see it happening. It's just a short little clip. Calcium will come into play. But cross bridges, where myosin attaches, it will attach, the myosin heads will attach to the actin. They'll zoom it even further. There's really long myosin heads attaching to actin, and they sit there and walk along, and they move the actin closer and closer and closer together. And I'm like, and so the whole thing shortens the muscle fiber. Yes, we'll talk about calcium in a little bit. There's your myosin head. It attaches to actin.